Hi, everyone, and I'm happy to present the first speaker of the VPH summer school, Miguel Ángel del Pozo. He first studied medicine in the University of Valladolid and graduated in 1991. After which, he specialized in immunology in the Hospital de la Princesa in Madrid. He did his PhD thesis in the Autonomous University of Madrid in biochemistry and focused on studying cell addition molecules and chemokines and their role in leukocyte recruitment during inflammation. Afterwards, he did his postdoctoral research in La Jolla, California, in the Scripps Research Institute in the laboratory of Martin Schwartz. There, he focused on the role that raw GTPases play in intercellular signaling, along with integrins and growth factors. So afterwards, in the same Scripps Research Institute, he became an associate professor with his own research group and continued focusing on the role of integrins, lipidic microdomains, and caveolae in intracellular signaling. In 2004, Miguel Ángel del Pozo received an award of Young Investigator Award, which is the predecessor of the ERC Starting Grant. And from 2007 to 2010, he has been the lead of vascular biology and inflammation department in the Spanish National Center of Cardiovascular Research. In 2007, he also received the Beckman-Coulter Award for researchers under 40 from the Spanish Society of Biochemistry and Molecular Biology. And since 2015, he has been a full professor in the Spanish Center of cardiovascular investigation. So he has made 146 publications to date and is currently the 8th most cited Spanish researcher in the field of cell biology. Welcome.
Shall I use this one? So thank you very much, Mane, for very kind introduction, and Jeroen and Annette and all the organizers for inviting me to be here. It's an honor to be here, and I hope that you enjoy this summer school. I think the program looks very multidisciplinary and very interesting, so I hope that you enjoy. So without further delay, I will start with my presentation. So we try to understand basically how cells adapt to mechanical stimuli, cells and tissues. So we are constantly surrounded by physical forces, and how are our tissues responding to that is our main question. So as this slide summarizes, this field is a blooming field in biomedical research. Not so long ago, there was not so many publications on this mechanobiology field, but as you can see, this has been growing exponentially, and it's even new terms such as mechanomedicine are being coined, showing how important this is for biomedicine. So the main principles of this field were shown by different groups. One of them is the group of Sirio Dupont and others, and in this group is in Italy, and they show that the same stem cell, in this case mesenchymal stem cell, when this is surrounded by a soft tissue, soft matrix, can give rise to an adipocyte. And the same cell, when it's surrounded by more stiff environment, stiffer matrix, it can derive into an osteoblast, completely different cell type. This was a landmark paper published by Sirio Dupont and Stefano Piccolo in Italy, and they showed that this was, only done by the surrounding matrix. How stiff was the matrix surrounding the cell? This is also very important for embryo development, and in cancer. In cancer, there was also another landmark paper by Valerie Weaver, which is in UCSF, in San Francisco, and they show in this case using in vivo with mammary cells, the same mammary cell with a soft environment is making normal acini. But if you increase the stiffness of the matrix with increasing concentrations of collagen, you can see how these mammary epithelial cells derive into aberrant morphology, and at the end you can even get a mammary ductal carcinoma just by the stiffer environment, showing how important this is in this case for cancer. In the institute where I work as Jerome Amman introduces the CNIC, National Center for Cardiovascular Research in Madrid. We are interested in this problem of atherosclerosis. I will talk a little bit at the end about this, because in our bloodstream we have different types of forces. You know probably better than me, laminar flow, oscillatory flow, and this disease, the atherosclerosis, which is actually the #1 killer, is responsible for ictus and for infarct, myocardial infarct. It only develops at places where oscillatory flow. So this is very important. In this case, the response of endothelial cells and smooth muscle cells in the vessels to different forces. Also, when you have an infarct, after this, depending on if you survive, how you organize the scar that is made from fibroblasts after the infarct, this is very important to allow that the patient will survive, basically, or not. In many diseases, fibrosis, in aging, stiffening for example, of the nervous central system is very important for dementia and for many things. So basically, we have this response in many diseases that we were not probably thinking about in the beginning. Again, this you know much better than me because you're a studying engineer. So this has been enabled by an interdisciplinary field in which physicists, engineers, and biologists and biomedical researchers are working together. I remember when I was doing my postdoc, I went to a Gordon conference in the year 2000, I think it was, and basically there was biologists and physicists talking like different languages. Nobody understood the other language. So this has been allowed in recent years because these physicists and engineers have developed, obviously not atomic force microscopy, but other, which was already there, but it's being used a lot for cells, and maybe you are doing experiments with this device, with atomic force microscopy, in which you can calculate the stiffness of the sample, in this case biological sample, but you can use optical tweezers, magnetic tweezers. We collaborate with a couple of groups here at the IBEC, Pera Roca-Cusack and Xavi Trepat, who are working, who actually developed this technique, and I will explain a little bit about this later. And also we collaborate a lot with Johann Guck in Germany, who developed this optical stretching and also real-time deformability cytometry, which is like a cytometer in which you can actually basically put forces to deform the cell and then measure the deformation of the cell. And this gives you a lot of information. So because of these techniques, now this field is progressing, but we still know very little about how different mechanobiology principles, I will say. For example, what are the mechanisms for sensing these forces? You have, for example, shear stress in blood vessels, stretching, which is sensed mainly by the smooth muscle cells in the case of the vessels. You have also different stiffness in the matrix, which is bound by these molecules integrins, which form these focal adhesions. But how are they sensing this force? And once the force is sensed, how is this transduced into the nucleus and into the different parts of the cytosol? How this force is transduced, how it is transformed into a biochemical signal, we don't know, or we know very little. And finally, how is the cell adapting or responding to these different forces? So at the end, because of this mechanical force, you have a different response in metabolism, in trafficking of membrane, in reorganization of the actin or the tubulin cytoskeleton. And also you have to integrate this response with the response to other stimuli, such as soluble factors, growth factors, cytokines in inflammation, and how are these different signals integrated? This we don't know. So the first question is how the cells sense this mechanical force, and this is where I will introduce these caveolae, which are invaginations of the plasma membrane, which we have been working with for quite a few years now. So these caveolae, I don't know if you are familiar with them. I don't know if you study these caveolae in the university. Do you study clathrin? You study clathrin coated tips. Do you know this? The music is not part of my presentation. Maybe to celebrate caveolae, but I didn't do it. So, an alarm or something. I don't know what is the time. Well, I will continue. So these are invaginations, nanoinvaginations of the plasma membrane, which are very small, 60 to 80 nanometers, which is a difficulty for us to study because you cannot see with regular microscopy, regular fluorescence microscopy. You need to use super resolution, which is still very hard, or electron microscopy. And they are very abundant in mechanically stressed tissues. As I already mentioned vessels, so endothelium, muscle, the heart. I will also talk about adipose tissue, which are also quite rich in caveolae, even though you might think that they are not stiff but soft. They are enriched in cholesterol and other lipids, and some of the main proteins making these invaginations are called caveolins, which are this molecule which is depicted here, which is in the inner side of the lipid bilayer in the plasma membrane. It's always bound to membranes. It binds cholesterol, as I mentioned, is actually the main protein binding cholesterol in the cell. And more recently, other proteins called cavins, which actually are the ones providing this electron-dense appearance to the caveolae, were identified. Among them, I will talk about this PTRF, also called cavin 1, which is actually related to transcription. It's actually called polymerase 1 and transcript releasing factor. So it's actually found in the nucleus or in the plasma membrane, which is a mystery how this might be happening. So I will be talking a little bit about this. Another important thing of caveolae is actin. So from the very first antibody that was made against caveolin, I think the paper was published in Cell around in the 1960s. A long time ago. So they found that they were following basically actin stress fibers. So it's associated to actin, which is already telling us a little bit about this mechanoadaptation function. And they are also very plastic. This, as you can see, they have different morphologies. It's not always the typical individual invagination, which are like these clathrin-coated pits, which are important for endocytosis. Sometimes they form these big vacuolae surrounded by caveolae, which are called rosettes, or clusters, which are typical in the muscle. And another important thing is that post-translational modifications of caveolin, the most important, or one of the most important, is phosphorylation of this tyrosine 14, which is done by the typical kinases of the SRC family, SRC, FIN, also ABL. And this is very important for most of the functions that were proposed for caveolae, such as endocytosis, remodeling of the matrix, migration. And this is why we made a mouse mutating this tyrosine into a phenylalanine. So we made a knocking mouse, which I will be talking about later. So the main function, as I mentioned before, it was thought to be important for endocytosis, for getting viruses and lipids and other things into the cell. But after many years of research, this was not very clear. And there was a couple of papers. One was published by the group of Pierre Nassois and Christophe Lamas at the Curie in France, and another one that we published the same year. And in this in this couple of papers, we basically established this mechanosensing and mechanotransduction principles of caveolae. So basically, when you have mechanical forces, such as shear stress or stretching or stiffness, what happens is that this invagination basically behaves like a spring, so it can flatten, it can change the morphology, and when this happens, the components dissociate. Caveolin is always bound to the plasma membrane. It cannot be in the cytosol. It will be because it's hydrophobic, so it has to be always in the membrane. And what happens is that these caveins then go to the nucleus and then they regulate transcription. And when this happens, if you have caveolin in the plasma membrane, you organize these previously called lipid rafts, maybe you're familiar with this concept, which are like heterogeneous domains in the plasma membrane, which are very rich in cholesterol. So they have this liquid order structure as opposed to this liquid disorder, which will be like the normal membrane. And in these liquid order regions is where caveolin is, because it's basically they are made by cholesterol, which is this tiny blue molecule here, the cholesterol, that intercalates into the lipid bilayer. And then caveolin is obviously enriched here and is sequestering this molecule, P190 Rho gap, which is a gap or an inactivator of the GTPase Rho, which I don't know if you are familiar with Rho GTPases. They regulate actin cytoskeleton. So basically the main function of this Rho is to trigger actomyosin contraction, which then in turn will activate the cytosolic site of the integrins, which then will be in active conformation, and integrins bind fibronectin, collagen, and other extracellular matrix protein. So what happens with all this pathway is that the integrins in active conformation are remodeling the matrix. Basically, they are making organized and stiff matrix. And this, we saw that it allowed the migration, for example, of tumor cells in a directional manner. If you don't have caveolin, what happens is that these liquid order domains don't have this protein. In this case, P190 Rho gap is sequestered into these domains, and this is actually where we also found Rho in the same domains. So then they are together, and they are P190 Rho gap is inhibiting. I don't know what I said before, but probably I said in the wrong manner. What happens when you have caveolin is that P190 Rho gap is excluded from these liquid order domains. So it's not in contact with Rho, so then Rho is free to activate actomyosin contraction. I think I said in the other way around. So what happens in caveolin knockout is that Rho is inhibited, there is no actomyosin contraction, the integrins are not active, and the matrix is kind of soft. It's kind of, if you like, Formula One. I think that yesterday you had the Montmeló, so maybe some of you were there. I am not a fan of this thing, but basically what happens is when they talk about the stiff or soft wheels, also the road itself, or maybe you can think also in the beach. When you go to the beach, when there is like wet surface, is stiffer than when it's not wet, and in the wet areas you can run. You can walk faster. If you go to the soft, this dry part of the sand, you cannot really walk in a fast manner. So this is what happens here. Tumor cells cannot really migrate, so they kind of stay in place. So basically, this is what we think is the main function of this caveolae: mechanosensing and mechanotransduction, organizing responses to mechanical forces. Another important thing of caveolae is that they are, as I mentioned, very plastic. Here, you can see the typical individual caveolae, one single invagination, but when we apply force, in this case, we trigger actin polymerization, so the membrane is constantly being stretched. And what happens is that you can see how these invaginations kind of flatten. In this case, we use immunogold to stain for caveolin. So you can, an electron microscopy, so you can see the wider neck in the caveolae compared to this one, and the presence of caveolin there. So this is kind of showing that this can actually flatten with mechanical force. So you have also these clusters and these rosettes, which are basically big parts of the plasma membrane with invaginate together, forming these vacuolae. So these rosettes were very intriguing, and what we did when Asier Echarri, who is now an assistant professor at the CNIC in the CIB in Madrid, when he was in my lab, he was interested in understanding how these rosettes, these big invaginations, responded to force. And what he found is that a molecule called FVP17, which is an F-bar domain protein, it's kind of like a banana shape, which you can see here. It's like a banana shape protein, which because of this form can bend the membrane. It can trigger membrane bending, and is responsible for the formation of these rosettes. So if we will knock out or knock down FVP17, we don't have these rosettes. And what happens in normal conditions with low, let's say with under basal, not high force, is that this FVP17, it has this name because it's forming binding protein, and it's binding a forming, which is called MDIA, a protein that is downstream of this Rho, which is responsible for the formation of stress fibers. So when FVP17 is in normal conditions, it's bending the membrane and is inhibiting MDIA, so then you don't have stress fibers. But if there is increased tension, what happens is that this molecule ABL, which is a famous tyrosine kinase which is involved in cancer, is the protein that is mutated in the Philadelphia chromosome in leukemia, for example, is phosphorylating FVP17. So when this is happening, is completely disassembling these rosettes, and then now MDIA is free to regulate stress fibers, so then it's increasing the tension in the membrane. So by coupling the membrane and the actin, these big rosettes are able to sense and respond to mechanical force. So what are the phenotypes in vivo of the absence of this caveolae? So you can do that with mutations of caveolin, for example, also with mutations in cabin. And there is actually mutations in human patients. And one of the main phenotypes is actually this lipodystrophy. These people cannot accumulate fat. So then you have increasing triglycerides and cholesterol circulating in blood, which is actually a really bad condition. So you might think that it's an advantage not being able to store fat, but at the end of the day, it's actually worse. It's triggering a metabolic syndrome, very similar to obesity. So they are very similar. If you don't have cabin, because cabin, or actually I didn't say this, but caveolin 1 is expressed in most tissues, apart from some types of muscle. And then you have other caveolin, which is called caveolin 3, which is very important for muscle. And then these cabins are expressed in all the tissues. So when you have caveolin 3 or cabin mutations, you have also different types of muscle dystrophy. So these are the main phenotypes of absence of caveolin or cabins in humans and also in mice. In mice is a very good model for studying lipodystrophy, which I will be talking later. So then, but basically what we found is that when you don't have caveolin, you have problems in many tissues, as I mentioned, all the tissues that are responding to mechanical forces. And I will be talking a little bit about some of these examples, such as, for example, in the adipose tissue. Okay, so another important thing of caveolae is matrix regulation. So I mentioned in this slide, which I was summarizing before, that there is a very strong link between caveolin and matrix, but by mechanical cues. So this is an example of this cancer situation which I was telling you before. So in this case, we have a tumor here. This T is the tumor. These are mammary tumors. And then this is the stroma surrounding the tumor. So the stroma, as you know, is made by fibroblasts, endothelial cells, immune cells. And in this case, we are using second harmonic generation. I don't know if you're familiar with this technique. You need like a 2-photon microscope. And you can visualize matrix structures that are very well organized, such as collagen. In this case, you don't see all the collagen, only the collagen that is making strong fibers or very organized fibers like these ones. So if you have caveolin in the stroma, the fibers are very strong, very parallel, and they are kind of perpendicular to the tumor stroma interface. And some researchers before showed that this phenotype is typical of invading tumors, such as I was saying before, with the that it will kind of allow the exit of the tumor cell from the tumor towards the stroma and then invading the tissues. If you don't have caveolin in the stroma, however, what happens is that you have less fibers, less organization, and they are kind of surrounding the tumor. They are not perpendicular. This behavior, this correlation of this morphology and invasion was established by Patricia Kelly in Madison University. So we actually collaborated with her to do these experiments. And then we did a very complicated experiment in vivo in which, I don't know if you can see, I hope you can see, in which what we did was to use a cell line which is called LM, which means lung metastatic. It's kind of like, it has tropism for making metastasis in the lungs, but actually, as you can see, they're making metastasis kind of everywhere. So when you inject in the mice this tumor alone, what you see is that there is not so many tumors. Here, there is a staining of a tumor, and in this case, in red, you see the fibronectin. Thank you. Yeah, I think you can see better. You can see the fibronectin in red, and then you have like the nuclei of the cells in blue, and then in this case it's a smooth muscle actin, which is staining for fibroblasts. In this case, we didn't inject fibroblasts, so then this is black. We only have we only have the tumor cell. So the experiment that we did to show if this was important in vivo was to co-inject this tumor cell line with fibroblasts. In this case, fibroblasts expressing caveolin. So if the fibroblasts express caveolin, you can see here, these are the fibroblasts, which are kind of elongated, and they are triggering this organization of the matrix. You have long and parallel and also stiff that we check by atomic force microscopy. So they are kind of very well organized matrix fibers. If, however, the fibroblasts that you inject are devoid of caveolin, then these fibroblasts are kind of not elongated. They are kind of round and they are not stretched. And then the matrix is very poorly organized. And here, the important slide, the important image at the end for my biomedical research is what is happening with the tumors. The same injection of the same number of tumor cells is making a lot of invasion and metastasis in the case of caveolin-positive fibroblasts, but not in this other case. So then the most difficult experiment came later, in which what we did was with this knockout fibroblasts, we also did like a trick, a genetic engineer trick, which was to inhibit P190 ROGAP, which is the one responsible for this inhibition of Rho. So if you inhibit P190 ROGAP expression, you don't have this, so then Rho will be free to maybe rescue this phenotype, and this is what we see. Now the fibroblasts are elongated again, the matrix is organized, and then you have more tumor cells. I have to say that actually this experiment was reproduced by an initiative to reproduce cancer cell papers from this is actually an old paper, 2011. So they pick our paper and they reproduce experiment, and we were successfully reproduced. So we can say that this is actually happening. The difficulty, because then we thought maybe caveolin is a target in the stroma, but it's very difficult to target the stromal cells. We have been trying, and then caveolin is doing some things in some tissues, some other things in some others. So it's very difficult to use it as a target, but maybe some of the things that it regulate caveolin or that are regulated by caveolin could be potentially used in therapy. And then I will summarize just other paper in which not only mechanically but also chemically we think that caveolin can actually regulate the matrix, and this was done by these exosomes, which I guess you study exosomes, right? In your degree in. I don't know if you study exosomes. They are kind of extracellular vesicles that are circulating, and they are made in this multivesicular body, which is a big endosome, very close to the endoplasmic reticulum. So what happens is that in this endosome, there is the formation of these intraluminal vesicles, which at the end are they are pinched off from the membrane of this endosome, and they are within the endosome. Then this endosome moves towards the plasma membrane, fuses with the plasma membrane, so then the vesicles, these exosomes, are released. And what we found is in caveolin 1, as I told you in the beginning, is very important to bind cholesterol. So basically, caveolin is binding cholesterol already in the ER and is trafficking towards the Golgi and towards the plasma membrane. So it's putting the cholesterol in the plasma membrane. So in normal cells, you have a gradient of cholesterol. You have a lot of cholesterol in the plasma membrane and much less in the nucleus or in intracellular vesicles. So in the case of caveolin knockout cells, because you don't have caveolin, cholesterol cannot get into the plasma membrane, so it's kind of enriched in these endosomes, in all of them. In the case of the multivesicular body, what happens is that increased cholesterol triggers an early pinch-off of these membranes, so they are very tiny, and then the exosomes, actually we measure with different techniques the size of these exosomes, and they were really small, really rich in cholesterol, and big proteins such as fibronectin or tenastin cannot get into the exosome because of their size, because they are like 200 kilodaltons, and they don't fit into these small vesicles. So this is very important because then the composition of the matrix changes. And then we actually measured that, and we found that apart from collagens are following the classical secretory route, which is ER, Golgi, trans-Golgi network, vesicles, and then released to the plasma membrane. This is the classical biosecretory route, and collagens are following this pathway. So they were known how they were deposited or secreted, but other proteins such as fibronectin or tenastin, not a lot is known about how they are deposited. So what we did in this plot is we are plotting the presence of certain protein in the exosome and what is happening in matrices which are derived from these cells. So basically, we take the cells, we let them grow for like one week or 10 days. They deposit matrix, then we kill the cells, we extract the cells, and we leave the matrix. And then we can do proteomics on this matrix. And as you can see here, when in the presence of caveolin, this is the knockout and this is the wild type, there is a strong correlation between the presence of these proteins in exosomes and in the matrix, suggesting then that these proteins are actually being released through this pathway. Okay, so another player in this field is JAP, which is a very famous protein. I'm sure you have been studying this JAP. So JAP is a master regulator of responses to mechanical stimuli. Again, this is the work of Stefano Piccolo and Cyril Dupont. And this is a review, or we adapted this from this paper, this scheme, in which basically what is shown is that this JAP is actually, it's not a transcription factor, but it binds to TEAD, which is a transcription factor. So it regulates transcription in response to mechanical force. So obviously for this, it needs to get into the nucleus. So when you have a soft matrix, or for example also with laminar flow, with a straight artery like the descending aorta, JAP is not going into the nucleus. You have most of the JAP, which is now here in green, in the cytosol. However, when you have a stiff environment, or also in this oscillatory flow, distal flow pattern in bifurcations or curvature of the arteries, now you have JAP imported in the nucleus. So then this is very important because when it's outside the nucleus, as I mentioned, is giving rise to an adipocyte, for example, osteoblast in the stiff condition, and the responses are basically different. Here you have low contractility, low remodeling of the matrix, and here you have high contractility and matrix remodeling, and also other responses such as increased proliferation or cell cycle stop in this other case. So this is very important. This is a very important factor, and as you will, maybe you have thought already, we saw that caveolin is very important to regulate this response. So in cells without caveolin, JAP is not getting into the nucleus, and this is a paper that Robert Moreno did during his PhD in my lab, and basically what he found is that this was related to actin, to something that actin was doing in this case. So basically he did an IP of JAP with an inhibitor of actin in cells with caveolin or without caveolin, and he found a protein called importin 7, which is here, which is one of the proteins that is very important to make other proteins such as SMAD3, which is downstream of TGF-beta, of MAP kinase 2, to be imported into the nucleus. So this is the normal function of this importin 7, what was known. And what was not known is that actually upon mechanical tension increase with, for example, a stiffer environment, such as here, with the stiffness of the matrix, what happens is that JAP is activated and is actually sequestering importin 7. It's like importin 7 will be like a taxi driver, and JAP will be the passenger, and the passenger is kind of sequestering the taxi driver and is out competing the other proteins. So then is being imported into the nucleus and then regulating mechanical response. So this was the thesis work of Maria Garcia in my lab with the help of Asier and Laura here. So then we are coming to the, I don't know how much, I still have time, right? Plenty of time. Okay, because we are not, I think we are kind of like in the middle or something. Okay, so this is basically the central hypothesis of our lab. We are trying to understand how is this response to mechanical forces, different types of forces, what are the properties of these domains that show this huge difference between high complexity when there is low force and very low complexity when there is high force. We are transitioning from very complex rosettes towards individual invaginations or basically no invagination at all. So how is this happening, and how is then this triggering this mechanical or chemical response, for example, in the matrix, which is important for normal homeostasis, but also in disease. So we are interested in different diseases, such as the formation of this atheroma or the tumor invasion. So basically, this is the current picture of the lab. So this is basically everybody that is in the lab now. And we have this central hypothesis. What are the mechanosensing and mechanotransduction properties of the caveolae, which are important for the adaptation of the tissue to this mechanical force. For example, we are focusing a lot on this remodeling of the matrix, which is very important. For example, in atherosclerosis, this is the atherosclerosis team, but also in immune, the arrival of immune cells towards the stroma, and how is this impacting, for example, the response to an infarct and also to a tumor. So then there is a part of the team which is studying the more basic responses to these forces. So I will be talking a little bit about the work of Fidel Lolo here, and then also in this mechanoadaptation, for example, I will be talking about the adipose tissue later in the talk. Okay, so the first question, very basic question, is how is this happening? How is this response or this sensing of the mechanical force happening? So for this, we have a couple of possibilities. One is that the whole invagination, the whole caveolae, important for the mechanical response, or whether it's just one of these proteins. In this case, we chose caveolin 1 because we have been working a lot with this protein. And basically for this, we have the first problem was that both proteins mutually regulate each other. So if you don't have caveolin, there is transcription regulation of cavin. So without caveolin, there is very little expression of cavin, and also the other way around. So the trick that Fidel did was to start from cavin knockout cells, in which, because of this regulation, there is no cavin, but also there is very little amount of caveolin. There is a little bit of caveolin, but very, very little. So in these cells, we then reconstitute the expression of caveolin. So we will have only caveolin. There is no cavin because this is a genetic knockout. Or if we reconstitute cavin or PTRF, then we have the whole structure, because also caveolin will be induced by cavin transcriptionally. So then we did a lot of experiments which are summarized here. So basically we did AFM here in Barcelona with Daniel Navajas and also with Jochen Gug then in Dresden. He now moved to another institute in Germany. We also teamed with Xavi Trepat and Pere Roca-Cusax here at IBEC in Barcelona using these magnetic tweezers to measure reinforcement, and we measure deformability with Jochen Gug. Unfortunately for us, when we compare the two cell lines, basically most of the experiments showed the same result. There was not a big difference, but we did see some difference to mechanoprotection. So this is the more basic experiment in which you basically do hypotonic shock. You wash the cells with a very hypotonic buffer, so then the cell swells, and if it is protected, it will stay, and if not, it will break. So these cells were showing a little bit more protection than these cells. They were breaking a little bit more with hypotonic shock. So then this was the most important experiment that we also team up here with a group here in Barcelona, maybe you know, Marino Arroyo, who is in the Barcelona University, and with his postdoc Nihil Walani, which is now back in India. So basically this is just mathematical modeling, and basically here is the situation of the whole caveolae, which are very stable. You will see that when you apply a lot of force, basically they will flatten, whereas these other guys, I don't know if you can see how they are responding to force gradually. So they are decreasing the height of this invagination gradually with force, whereas this guy is very stable. Basically, there is like a threshold behavior here, hypothesized by the modeling, in which it will flatten only with a high force. So this was suggesting that the critical experiment that we should do was optical tweezers. So in this case, basically you pull a tether of the lipid membrane, and then you swell the cell with hypotonic buffer, and you measure the response to this increased tension. So basically, this is what is being done here. And in the case of PTRF knockout cells, the cells didn't buffer this mechanical stimuli. So basically, they are increasing a lot the mechanical tension in this membrane tether that we are pulling. In the case of the full caveolae structure, they were not increasing. This was meaning that this membrane has buffer ability, so they don't increase the membrane tension a lot, correlating with more protection. So the cells, actually, we knew that they were dying much less than these guys. And as I mentioned before, also in mechanoprotection, these cells were like an intermediate behavior, which was also being seen here. In this case, we have like an intermediate increase. So they have like some buffering ability, but not complete buffering ability. Okay, so this was telling us that there was something there which was different. So we started a quest to find different type of invagination. And finally, we teaming up with Britta Qualman. This took us a really long time. So she's using a very interesting technique. It's called freeze fracture replica immunolabeling, which is like an electron microscopy, but very particular type of electron microscopy, in which basically you freeze the cell and then you break it, and then you take one replica of the plasma membrane, and then you look with electron microscope. And here you can see that in the caveolae normal situation, we see the classical invaginations, which are very tiny, 60 to 80 nanometers, and they are full of caveolin protein. This is these dots here. In the case of cells with only caveolin, we see much bigger invaginations. Actually, we see many different sizes, but from very big to very tiny. But we see the particular difference is this, or the specific difference is this very huge invagination. It's like a sinkhole, like a big depression in the plasma membrane. And they have caveolin, but much less caveolin than these other guys. So basically, maybe I didn't tell you in the beginning, this caveolae is getting the name from the Latin cave, which means cueva, I don't know, in caveolae, cave. And in this, we needed to give a name, and we came with this name, doline, which is actually coming from Slovenian language. So in the Balkans, you have this karstic phenomena in which they have like big depression because of the type of rock. And then we have in Spain, in northern Spain, we have this La Gran Dolina in Atapuerca. So it's basically like a big sinkhole a big depression. So then we gave this name. And this is like a 3D, we should see here a 3D reconstruction of this new formation that we identify, which is, as you can see, much bigger than caveolae and with very little amount of caveolin. Some caveolin, but very little amount. So these are the dolines. Basically, we did some functional experiments to see whether they were different, the response. So basically, what we did here is the quantifying the number of dead cells in response to different hypotonic challenge. So here is basically we are diluting the media, here very little, 1/3, 1/4, 1/5, and at the end 20 times the dilution. And as you can see here, the cells with caveolae are better prepared, as we knew already, for this challenge. So there are dead cells, but much less than in the other conditions. And these are the cells with caveolae. However, what the interesting thing is that when we were doing like intermediate dilution, the cells with dolines were actually better prepared than cells with caveolae. So basically, cells with caveolae are not responding to low dilution. They are dying basically the same as unprotected cells that don't have caveolae or dolines. And we also did a fancier experiment looking at JAP localization in the nucleus. So this was done with a high-throughput microscope called Opera, in which you can take many images. You basically do like a high-throughput scan of the sample, and we see like we have like a small objective like 10x or 20x, and you can see the localization of the nucleus of JAP in the nucleus. I hope you can see. And here is the small dilution. And what you see is that in the cells with dolines, in this condition, with very little dilution, you start to see JAP in the nucleus, which you don't see in the PTRF plus PTRF, the PTRF knockout plus PTRF in the caveolae cells. In these cells, you need like a bigger dilution to start to see JAP in the nucleus. So then, also at the level of mechanotransduction, the JAP nuclear import, we see differences in these two conditions. So basically, we can propose a model in which we will have two different systems to adapt to mechanical tension. This is what was known before, the caveolae, which is very rigid structure in the plasma membrane because of the presence of these cabins, which are making like a shield in the membrane, resisting deformation. So you need basically a threshold. So for the cabins to dissociate from caveolin, they are normally binding. Cabins are binding caveolin and also lipids in the membrane. So when you, after this threshold, you have basically like a switch behavior, switch off, switch on. It's like a qualitative difference. You have either no response or response after the threshold. However, in the case of the dolines, we are constantly responding to, when you increase membrane tension, we are having like a constant response. And basically the model, I will show this now in next slides, what is predicting is that this, instead of flattening, the way to respond will be like a splitting the membrane in different dolines, because we were, as I mentioned, we were finding dolines of many different sizes, very big, but also not that big. So, okay, so we published this in Nature Cell Biology recently, and this is the model of this splitting. So when we increase, in the case of here is the case of the caveolae, in which you either have caveolae, and when after the threshold you have the disassembly of the caveolae in the presence of PTRF, but when there is no PTRF, when there is only caveolin, in the red behavior, basically what the model was predicting was that with increased tension, there will be like a splitting of the domain into smaller domains. So to test that, it was very difficult for us to do it live with stretching, even though we are now trying to do. But we did a trick, which was to use cyclodextrin, which is a drug that is basically sequestering cholesterol. And when it sequesters cholesterol, it's increasing the tension, because cholesterol is providing, by intercalating into the lipid membrane, is providing this buffering ability. So I hope you can see now, if you look at these movies, which are in which we have caveolin labeled with GFP, so in green, when we give cyclodextrin, that you will see like a splitting of one dot into other dots, like this in these cases. For example, here. We also see some splitting behavior in this. I will play it again. See, hope it works. Okay. So we also see some here, but much less. Because so why is this happening? As I mentioned, when we do a PTRF knockout, there is transcription regulation. There is decrease of caveolin, but you still have some caveolin. There is not complete absence of caveolin. Actually, we find some dolines, much less, but we find some dolines in this condition. So we can actually quantify this. So this is the splitting behavior, like in which one dot is being split into more dots. So this is quantified here. So then we did some biochemistry to try to characterize these domains, and this was established before by Ari Helenius, a classical researcher in caveolae, which was working in ETH in Zurich, and who is now actually retired, but was one of the pioneers in caveolae. So basically, he was doing sucrose gradients, in which you can lyse the cell and then do like a gradient of less an increasing amount of sucrose. So then you will have like the splitting of the caveolin domains into different aggregates, because caveolin is actually binding itself. Caveolin 1 is binding to caveolin 1, forming oligomers, and it can also bind to caveolin 2, forming like many different sizes of oligomers, in which so then there is a very big complex which is called 70S, which it was postulated as the caveolae, but also this researcher was finding this 8S much smaller complex. So also this is this 70X big complex is sensitive to cholesterol depletion when we use this drug, cyclodextrin, which is the drug I was using before in these videos to sequester cholesterol. Basically, is a dextrin that has like a hydrophobic pocket which is sequestering cholesterol. So when we deplete cholesterol, this 70S basically disappears. And this is the caveolae positive cells, PTRF knockout plus PTRF. In the doline cells, what is happening is that we only find this 8S complex. We don't find the big one. And it's actually this 8S complex, we see it like in the control situation, that is actually resisting cyclodextrin treatment. So this is very interesting because it has been published recently. A potential structure of this caveolin of this caveolae, in which like a disk of different amounts of caveolin. In this case, they postulate a number of this, which is not what we find. We find something a little bit different. But we think that this disk of caveolin will be probably these dolines, this 8S complex that we are now studying. We are doing proteomics, lipidomics. We are trying to work with the structure of this domain. So this is an ongoing work in the lab. Okay, so I'm coming towards the end. When is it's like till 11 is when we finish, right? At 11, okay. So I think it's like 10 more minutes or something, and then we have time for discussion. So in this last part of the talk, I'm coming back to this slide that I showed you, that in the soft matrix, these mesenchymal stem cells can differentiate into an adipocyte, very different to what happens only when you change the matrix. For example, you increase collagen or fibronectin, you have a stiffer matrix, and then you have an osteoclast. An osteoblast, actually, sorry. So in this adipocyte is a very interesting cell because it's basically one big lipid droplet, which is basically a tiny nucleus, and the rest of the cell is a big vacuole, which is called lipid droplet, and this is our adipocytes. So as I mentioned in the introduction, one of the main phenotypes of caveolin knockout mouse, but also in patients, is that they cannot store fat. So this is an NMR image in which in white you can see the fat deposit. Here you have to, I hope you can see that this is like a normal patient, normal human, so then, or wild type if you want. So then you have fat storage. But in these patients that have these mutations in caveolin, you cannot, if you really increase the contrast, you see the bone, but you don't see basically fat deposit. So this is very interesting. And why is this happening? We studied metabolism of these patients, and actually they have problems, but especially the problem is dyslipidemia. So they have increased cholesterol, very high levels of cholesterol and triglycerides in the blood, but they don't store it in the white adipose tissue. So the hypothesis here is that basically it could be also a mechanical response, so that when you increase the amount of fat in the normal adipocyte, you will have like healthy obesity, normal metabolism, but you can have also a situation in which you cannot store fat just by mechanical problems, and then you have this lipodystrophy. So this was the hypothesis, and obviously our hypothesis is whether it was caveolin dependent, because there were like 3 papers showing that caveolin was very important for lipid droplet formation and is very important in the adipocyte. And for this, as I mentioned in the beginning, we also used this knock-in mouse that we built in the lab by basically Marta Guadamillas and Ana Cerezo. And so basically they mutated this tyrosine, which I mentioned was very important for response to the mechanical, was very important for in vitro functions of caveolin, such as endocytosis or matrix remodeling. So we mutated into a phenylalanine, so this amino acid cannot be phosphorylated, and we did this mice. And this was important because, in this case, because we knew that these mutant mice can make caveolae, so caveolae apparently are normal, but they cannot be endositized, they cannot be internalized, so there is a delay in the internalization. So we were studying the 3 genotypes, wild type, knockout, and this Y14F mutant. And the first experiment that we did was in collaboration with Ricardo Garcia, who is an expert on, and Carlos Guerrero, he's a PhD student at the time. He's an expert on AFM, and basically we were measuring in adipocytes that we were extracting from the mice. So we were making like tissue explants of this fat. And Maria Boyd, during her PhD, was measuring the stiffness of these tissues. And as you can see, there is a huge increase in stiffness in the case of caveolin knockout. And this was mice that were subjected to this high-fat diet, so they were really obese mice, and for 45 weeks, which is here. This is an image. And there was a problem because we realized that actually in this situation, you have increased fibrosis, which you can see here, and also the presence of some immune cells which were infiltrating the adipose tissue. So there was already like obesity, metabolic syndrome, inflammation. So we thought maybe they are stiffer because they have fibrosis, so then this fibrosis is making it the whole thing stiffer. So to rule out this possibility, what we did was to do the same experiment in very, very young mice, only 5-week-old, and with normal diet, and we saw the same behavior. So there is already increase, and in this case you don't have inflammation or fibrosis. So it looks like it's not secondary to the fibrosis, but it is happening before the fibrosis. And also, what Maria Boyd did, she was very organized and very consistent in her research. So what she did was to do the same experiment in vitro on adipocytes which were isolated from the mice in culture. And also you can see an increased stiffness in the case of the knockout. And in the case of the mutant, which I mentioned, they can make caveolae, they had like an intermediate phenotype. So we took a look at this caveolae, and this is a summary of many experiments. And what we are doing here is to increase the size of the lipid droplet by giving the adipocytes oleic acid, which is a lipid that is being stored in the adipocytes, and we were comparing the wild type and the mutant, that both of them can make caveolae, as you can see here. So when there is little amount of this oleic acid, the lipid droplet is very, very far away from the plasma membrane. So here we are measuring the plasma membrane to lipid droplet distance in here. And in this case, we are towards this, we are increasing the amount of oleic acid. So then the lipid droplet is growing and is coming closer to the plasma membrane. So when this is happening, as you can see, in both situations, there is a decrease in the number of caveolae. So in the case of the wild type, when the lipid droplet grows, the lipid droplet is here. This is one cell and this is another cell. So these are two different cells. The lipid droplet is growing and then caveolae basically are disappearing, which is the green bars. Okay, so, and the mutant is also decreasing the amount, but much, much slower. And especially when you are very close to when the lipid droplet is very close to the plasma membrane, is when we have more difference, as you can see here, compared to the wild type. And the important thing was to look at the caveolae itself. And when this happened, we saw that the caveolae were no longer these spherical, kind of spherical structures, but they were more elongated. So it's like they have a difficulty in the flattening. So this is like a scheme in which when we increase the size of the lipid droplet and it's getting close to the plasma membrane, in a wild-type situation, you have this tyrosine 14 here, which is this positively charged residue. When you have increased amount of fat, basically the caveolae flatten. And obviously there is a role of this tyrosine within, because without the ability of being phosphorylated, they didn't flatten as well. So we did a number of experiments. I'm not going to get into the details because I think they are very specific and I want to leave more time for discussion. But basically we did some mechanistic experiments to try to understand what was the role of this phosphorylation. And basically these are these gradients that I was mentioning before, in which we are doing oleic acid loading. And this is basically the quantification of this. So what we have here is in this line here, we have the top of the gradient, and this is basically lipid droplet presence of the protein in lipid droplet. This is a control perilipin 1, which is a protein that is always in the lipid droplet. But basically, when we increase the amount of fat, when we increase the size of the lipid droplet, there is a transfer of caveolin from the plasma membrane towards the lipid droplet. Also, another component of the lipid droplets of the caveolae, which is called AHD2. But for example, cavin is not being transferred because there is already some cavin in the lipid droplet, and this is not increased, which is quantified here. So this is control, and with oleic acid in the wild type. Obviously, in the mutant there is no change, and in the wild type there is increased caveolin and AHD2 in the lipid droplet. So it's kind of with phosphorylate, with increased tension, which is triggered by this, we see phosphorylation, which I didn't mention, but we saw here with the increase in the same experiment, there is increased phosphorylation of caveolin with increased tension. Okay, so then we saw a defect in the oligomerization of as I mentioned, caveolin can form dimers and very big oligomers. And in this mutant, there is increased dimerization and oligomerization, meaning that the caveolin is not assembling properly. We also did some TIRF experiment in which we can see the novosynthesis of caveolin, and we always see much more amount in all conditions, of caveolin in the mutant, which is similar to the electron microscopy images that I summarize here, in which we always see a little bit more amount of this caveolae. So it's like maybe because it's not functioning properly, it's not being assembled properly, the cell is trying to make more caveolin in the case of the mutant. And also I don't know if you're familiar with this probe, it's called Flipper. So Flipper is a probe that also intercalates into the plasma membrane and can actually measure tension. So when we increase the oleic acid loading, in the case of the wild type, we see that the cells which are starting from an increased flipper, which is kind of like a stiffer membrane, is decreasing this tension. The tension is being reduced when you increase the size of the adipocyte with this oleic acid. And in the case of the mutant, however, is starting from a lower amount of tension and then it's increasing the tension. So then it's kind of not buffering the increase of the size properly. So this is also reminiscent of what I saw, if you remember, for the AFM, in which this mutant has increased stiffness compared to the wild type. And at the end, we did FRAP experiment in which we also increase the size of the lipid droplet. This is the blue curve. So in the case of the wild type, when you increase the size, there is like more release of proteins, which we think is the caveolin that is being released from the caveolae. And in the case of the mutant, is the other way around. When you increase the tension, it's kind of you have less release of caveolin or less diffusion of caveolin. So this is the model that we have. There is normal synthesis of caveolin in the wild type. When there is tension increase, there is flattening of the caveolae and transferring of some proteins towards the lipid droplet. So there is like an exchange of proteins from the lipid droplet towards the plasma membrane. And in the case of the mutant, there is more caveolin being synthesized. There is defect in this release of the protein when you increase the tension, and there is this pool of oligomers which are not being normally released from the caveolae, forming another domain which we are trying to characterize in the case of the mutant. So basically, we have when there is an increase of the amount of fat, which is normally stored in an adipocyte. We still don't know how is this happening, because it's possible that cytoskeletal differences, but these cells have very little amount of actin polymerization, because basically everything is lipid droplet. So we are leaning towards a hypothesis in which probably this repulsion of the caveolin being present in the lipid droplet, because they will have positive charge, maybe because of this repulsion, is facilitating the flattening of the caveolae. And in the case of the mutant, because there is no transferring of caveolin towards the lipid droplet, and there is no difference in charge because there is no phosphorylation, maybe there is a defect in this flattening. So this is the summary of the talk. As I mentioned, we are trying to understand what are the response, how is the response of the cell to different mechanical forces, including shear stress, stiffness. How are these domains responding to different mechanical forces? So we introduced this new domain called doline, which we think is very important for response to normal forces, because in tissues you can have very high force in some situations in which you require this flattening of the caveolae, but normally we don't have very high forces. We have much lower forces in different situations, and we think that probably dolines are responding more to these small and medium forces as compared to caveolae. In these complex functions that we are trying to understand for this mechanical response, we also introduce how YAP is being imported into the nucleus upon mechanical tension, the role of these exosomes in changing this matrix composition, and what the behavior in these adipocytes. And just finally, I just wanted to mention maybe a question for you for the discussion, which is what will be because I work in a cardiovascular center, and I didn't talk about this. Actually, lipodystrophy is very important for cardiovascular metabolism. But the question is, in our circulatory tree, let's say, we have these bifurcations, ramifications, and despite the fact that cardiovascular risk factors, like smoking, high-fat diet, sugar, not doing exercise, aging, I mean, all these factors are systemic, so they will act in all the points of the cardiovascular tree. But we only have the deposit of this atheroma plaque in these inner curvature regions or in these bifurcations. The difference in these areas is basically flow. So in straight areas, we have laminar flow, which is a very, very fast force, only in one direction. And in the case of the disturbed flow areas, we have very low shear forces and in multiple directions. And basically, people have shown how in these areas you have increased inflammation, increased oxidation, ROX production. the recruitment of immune cells, and basically the opposite in the laminar flow areas. So we are trying to understand how is this happening, and what we are doing is basically trying to understand the first step of this, which is basically the activation of the endothelial inflammation. And for this, we did some screens already in cells subjected to disturbed flow. And we have found that there is like some genes which actually activate TGF-beta, which is mechanical remodeling. Some genes which are basically lipid metabolism, but also the pathway that activates NF-kappa B, which is basically inflammation. What we know is that this changes the composition of the matrix to make possible the retention of the LDL and then the recruitment of these leukocytes. And what is happening in atherosclerosis is that this is basically creating positive feedback loops, which makes this area even more sensitive to this disturbed flow. So basically, this is the purpose of a Lacaisagram that we are now trying to do. So basically, this is the lab, and I want to thank the people that were involved in the different projects. Fidel was main responsible for the Dolim project, which I talked about. Maria Boya and Marta Guadamillas were involved in the adipocyte story, and many people here are working on different projects. And also want to thank collaborators, which I mentioned during the talk, and funding. And just to say that we have postdoc positions available at CNIC if you're interested. We also have some PhD positions, and these are just the images of the lab showing that we also do celebrate some things. So thank you very much for your attention, and I hope you have many questions.
Anybody has questions?
Thank you, Miguel Ángel. It was a very fascinating talk. In the part where you were talking about the caveolae and the stretching, when they are submitted to mechanical forces, you mentioned that the integrins realign or they can realign the extracellular matrix. How did that happen? I mean, I don't know. Perhaps you explained it and I missed it, but.
Yes, this is not our work. This is based on the. I mean, what we find is actually the alignment of the matrix. Mm-hmm. So we did that in different ways. Before, basically, the first experiment was just to stain for collagen or fibronectin, and we were actually measuring the lines with different programs. You can then, what we did was to calculate the angles in which they were crossing. So then, basically, in the wild-type situation, there is like more parallel, and in the knockout, there was more like chaotic or isotropic behavior. So then we did AFM, and the wild-type were like 40% stiffer than the knockouts. And then it was known that, I mean, all the other elements were kind of reported. We reported the caveolae impingement interrogap to raw regulation. So then, in the case of the knockouts, I didn't show again, but the fibroblasts are like really, well, the wild types first, they are like polarized. So there's like a leading edge, they have stress fibers, and then they are like migrating in one direction. In the case of the knockout, they are completely round, like a fried egg behavior. You have like stress fibers which are like concentrical. They cannot make big focal adhesions. They make small focal complexes, but because there is no actomyosin contraction, there is no formation of big focal adhesions. So then the integrins, we knew, also this is like a previous paper that I didn't have time to show, but we know that the integrins are much less active. So you can do this with different antibodies. 9EG, you probably know because of your work. 9EG10, this different HATS21, they are decreasing in the case of the caveolae knockout. So then the integrins are like this the closed conformation. The active is the open extended form. We haven't looked at the integrins by this in the knockout, but we think that they are in the by the antibody experiments, they are in the non-active conformation. So we think that this is what is explaining the remodeling.
Okay. Thank you.
Sure.
Thank you very much, very impressive work. And I have a question about these new structures, dolines.
Yes.
Do you have some idea about, because I understood that the presence of dolines allow better adaptation to mechanical stress. Do you know in the cardiovascular system how there is the distribution of these dolines, and if they are more in the areas of higher stress?
This is a very good question. And this is what we are doing. Like, the work of, Celia, her name is here. I don't want to. So Celia is just starting her PhD on exactly that question. So she's working together with Fidel. Actually, Fidel is her co-PhD director. So it's very difficult to see dolines, but we are trying to see. Our hypothesis is that in these atero-prone regions where you have the disturbed flow, because there is lower forces, so then dolines will be enriched. So we got some images like recently from Brita Qualman, but we are trying to establish this in Madrid, this technique, and we see some dolines, but we still cannot say for sure, they are more in the atero-prone compared to the atero-resistant, or laminar versus disturbed flow areas, but this is the hypothesis. Actually, we only have one paper on dolines, but we think that in regions where there is like very low forces like disturbed flow, or maybe, liver or brain where there is like very low force, and also it fits with the fact that neurons have very little amount of caveolae and basically no caving, that it could be also there. So then we are also looking in tissues like this.
Thank you very much.
Yes.
Interesting.
I have questions. Yeah, it's also about dolines. So does dolin splitting further lower the threshold of mechanical force that the cells respond to?
Sorry, can you, if the split?
Yeah, you described dolin splitting.
Yes.
So does the splitting further lower the threshold of mechanical force that the cells respond to?
Yeah, this is a very good question. That this splitting, because it's kind of adapting to the increased tension, it will change the threshold. We don't know. I mean, we would love to be able to address that question. Maybe teaming up with some engineer. But this is actually, I think it was predicted by the model. But I have to check with Marino and Miguel. But basically, my expectation is yes, probably this is what will happen.
Yeah. Okay. Thank you so much.
And actually, maybe a hint towards that is that we actually find that cells with can have both dolin and caveolae. So then probably, yes, but it will be very nice to find out.
Yeah, it would be also very interesting to see how this distribution of dolins and caveolae affects gene expression and cellular phenotype in general.
Yes.
Thanks. Yeah, I have my mic. Yeah, thank you very much. Actually, I had a question a bit so in that direction. So I was wondering whether you have an idea of the regulation pathways that are differentially activated when you have then dolines or caveolae with a differential regulation of CAV1. So the idea is that there may be like a natural switch between one very specific mechanotransduction pathway adapted for relatively low mechanical forces, and another mechanotransduction pathway that would be triggered with high forces.
This is the idea, but we know very little. The only thing that we know for sure is that at least they are able to mechanotransduce, because we look at YAP, and we have a response in YAP, which is not happening in caveolae till very high force. So, but we don't know what is the mechanism, how this is happening. And this is a very important question, because also in the field of caveolae, if you look at all papers, basically, most of the papers were about signaling because, you find Rob Parton and John Hancock in Australia. This is like work in the 90s, that there is a difference in HRAS, KRAS signaling. The way I came into this was from Roger TPS as I was doing my postdoc in RAC, and we found that, I didn't mention that, but part of the phenotype of the caveolae knockout is that RAC is starting the signaling from the plasma membrane because there are plextrian homology domains in GEFs which activate RAC, which are in the plasma membrane. So when you don't have caveolin, because there is no internalization of this domain, they are always in the membrane, so then RAC is always active. And then this is also explaining why they are round, because not only because they have, like I was saying before, this very low rho, but they have high RAC, so then it's making protrusions in all the plasma membrane. So they don't have like persistent migration. They are migrating like nowhere. So then this hints towards, this was caveolin or caveolae dependent. It was doline or, so there are many experiments that we need to do. But my hypothesis is that, yes, probably they have just caveolin doing some things without the invagination. The other thing is that the tissue, heterogeneity, like in different cells, you have different receptors. You have caveolae in many different types of cells. So this is what I said, that we have like a different response, completely different in different tissue. And, so this makes it quite complex to address. But,
and then another question, maybe. Is there questions here? No? So
you can tell them that for this price of the hands-on, maybe if they ask, you can give them 10 points or something.
I was considering that. You haven't mentioned at all the adherence, the gap junctions. So do you have actually, do you know if there is a relation between then the regulation of these cell-cell interactions and the regulation of the caveolines? Or
I get this question in, yes.
Signals that would come from the gap junctions or the adherence?
Most likely, yes. But, in epithelial cells, there is like an increased trafficking of caveolin towards the apical compared to the basolateral. So probably yes, it depends on the cell type and on the context, but we don't have any data to support that. But when I go to places where they work with development and blood vessels and they have this type of question, yes.
Okay, so who is doing the hands-on on biodynamo? All right, so keep in mind on the caveolin aspects. All right, so, well, if there is no further question, so we're on time then for the coffee break. Miguel, Ángel, thank you very much.
Thank you very much.